The remaining part of the carbohydrate metabolism, that means the regulation of carbohydrates, and in lecture 11. So this is the pathways of glycolysis and gluconeogenesis, where the three irreversible steps of glycolytic pathway catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase will be the enzyme of interest to regulate the glycolysis. And similarly, the three irreversible steps of gluconeogenesis, like the steps catalyzed by carboxylase, pyruvate carboxylase, fructose 1,6 bisphosphatase, and glucose 6 phosphatase, will be the enzyme of interest to regulate the gluconeogenesis. So what is about the phosphofructokinase 1? This is under allosteric regulation. So this is the phosphofructokinase 1. So the irreversible reaction catalyzed by phosphofructokinase 1 is the step that commits a cell to the passage of glucose through glycolysis. This enzyme has the binding site for the substrate fructose 1,6 phosphate and also ATP. In addition to the binding site for these two substrates, this complex enzyme has several regulatory sites for allosteric activations and inhibitors. So wherein high ATP levels signal that the cell is producing ATP faster than it is consuming, then ATP inhibits phosphofructokinase 1 by binding to an allosteric site and lowering the affinity of the enzyme for its substrate fructose 6 phosphate. So citrate, the ionized form of citric acid, and is a key intermediate of the aerobic respiration of pyruvate. That means pyruvate to acetyl-CoA and acetyl-CoA to the TCA cycle produces citrate. So citrate is the first intermediate produced in the TCA cycle upon arrival of acetyl-CoA. So where acetyl-CoA react with oxaloacetate to form citrate. So citrate is a very important molecule that serves as an allosteric regulator of phosphofructokinase 1. So this accelerates the rate of ATP to inhibit the phosphofructokinase 1. So higher citrate concentration increase the inhibitory effect of ATP and further reducing the flow of glucose through the glycolysis. The most significant allosteric regulator of phosphofructokinase 1 is fructose 2,6 biphosphate, which strongly activate the enzyme. So here is the schematic diagram of phosphofructokinase 1 and fructose bisphosphatase 1, how they are regulated with how they are regulated by ATP, citrate, ADP, AMP. Okay, similar like that. This image just simplified image of the bisection of glucose to enter into gluconeogenesis or glycolysis. So let's study the regulation of gluconeogenesis. So the first enzyme that is involved in gluconeogenesis is pyruvate kinase that catalyzes the pyruvate to form oxaloacetate. So here the most important anaphylaxis reaction in mammalian liver and kidney is the reversible carboxylation of pyruvate by carbon dioxide to form oxaloacetate, and catalyzed by pyruvate carboxylase. This is the first step of gluconeogenesis, and gluconeogenesis is regulated at the level of pyruvate carboxylase enzyme. So pyruvate carboxylase also play an important role in the formation of myelin sheath that surround the certain nerve cells and the production of neurotransmitters for communication between neurons. Pyruvate carboxylase deficiency is a rare genetic disorder present at birth. So what happened here, if there is a lack of pyruvate carboxylase enzyme, or the deficiency of pyruvate carboxylase enzyme will switch off the conversion of pyruvate to oxaloacetate and followed by phosphoryl pyruvate. That means it inhibits the first step of gluconeogenesis. So if the gluconeogenesis is stopped, then the accumulation of pyruvate will be observed. So accumulation of pyruvate will lead to the formation of alanine in the muscle. So we know pyruvate takes the amino group in the muscle and convert it to alanine, and this is the non-toxic form of amino acid transporting system or amino acid transporter and throughout the blood. So if the pyruvate is accumulated, the concentration of alanine will be increased, which leads to the conditions called hyperammonemia, and also increased concentration of pyruvate will produce lactate. So this is also a consequence of the lactic acidosis. This is a very abnormal conditions for the body. Okay, and also if the concentration of pyruvate is increased, or if the pyruvate is unable to introduce into the gluconeogenesis, so pyruvate will be converted into acetyl-CoA. So acetyl-CoA will then further convert it to fatty acid or acetoacetate, which leads to the formation of ketone bodies. So these are three important consequences if the pyruvate carboxylase enzyme is deficient. So what is in general, they also the accumulation of lactic acid producing lactic acidosis may then lead to the liver failure, hepatomegaly, demyelination of neurons, and also hyperammonemia result from poor ammonia disposal and decreased urea cycle function. So children with pyruvate carboxylase deficiency usually die So children with pyruvate carboxylase deficiency usually die in infancy or early childhood, but some survive to adulthood, having decreased muscle tone, hypotonia, intellectual disabilities, abnormal eye movements, irregular sign and reflexes due to damage of upper motor neurons by pyramidal tract sign, seizures, and coma and other conditions. So fructose 1,6-bisphosphatase, this is the second enzyme of enzyme that regulates the second irreversible reactions in gluconeogenesis here. So fructose 1,6-bisphosphatase deficiency is an inherited metabolic disorder in which the blood cannot properly make glucose. So without effective gluconeogenesis, hypoglycemia will set in after about 12 hours of fasting, and this is the time when liver glycogen store have been exhausted and body has to rely on gluconeogenesis. So high level of glucagon will tend to release fatty acid from adipose tissue in that time. And this will combine with glycerol that cannot be used in liver to make triacylglycerol, causing fatty acid. So what, in general, the deficiency of fructose 1,6-phosphatase will inhibit the transfer of fructose 1,6-phosphate to fructose 6-phosphate. That means this enzyme deficiency leads to the stop of gluconeogenesis. So due to the deficiency of this enzyme, gluconeogenesis will completely stop. So as a result, this will produce hypoglycemia when all the glycogens in the liver will exhausted. So at the same time, when blood glucose level is dramatically reduced, the hormone glucagon will be released, and which will catalyze the breakdown of fatty acid or fat from adipose tissues, which will then combine with glycerol in the liver and make triacylglycerol. That means decreased blood glucose level will tend to release glucagon, and glucagon will break down the triacylglycerol in adipose tissues. Those triacylglycerol will form fatty acid, and this fatty acid will combine with glycerol in the liver and will make triacylglycerol and deposit in the liver, which leads to the development of fatty liver. So inhibition of fructose 1,6-phosphate bisphosphatase can lead to the hypoglycemia and can lead to the development of fatty liver. So without another case, so if this enzyme is switch off or if this enzyme is deficient, the ATP will not be produced here. So without ATP will not be breakdown here in phosphate. So without phosphate here, ATP will not be converted. Look at here, if fructose 1,6-bisphosphatase is converted to fructose 6, one phosphate group will be removed by the breakdown of fructose 1,6-phosphate. So if this enzyme is deficient, the phosphate will not synthesize. If the phosphate is not synthesized, then ATP cannot be made, and many processes cannot occur. So as 3 carbon molecules cannot be used to make glucose, they will instead be made into pyruvate. So if this fructose 1,6-phosphate cannot be converted to fructose 6-phosphate, so it will be converting this one and finally pyruvate. And if pyruvate is produced, this will be converting to lactate. This acid causes a drop in the pH of the blood, and acetyl-CoA will build up, leading to the creation of ketone bodies, similarly to this slide. So here are the consequence of the deficiency of the last enzyme glucose 6-phosphatase, which catalyzes the last reaction of gluconeogenesis. Here, glucose 6-phosphatase deficiency is a group of inherited metabolic diseases characterized by poor tolerance to fasting, growth retardation, and hepatomegaly. So if this enzyme is inhibited, glucose 6-phosphate will not be converted into glucose. So it's the glucose concentration in the blood will be decreased. So and glucose will not be synthesized from non-carbohydrate source. So if in the body the glycogen is completely used up, so no glucose will be formed because of the lack of glucose 6-phosphatase. And previously we studied fructose 1,6-bisphosphate and pyruvate carboxylase. So in that time, this will be very difficult for the person to have a fast. The deficiency of glucose 6-phosphatase will be characterized by poor tolerance to fasting, growth retardation, hepatomegaly resulting from the accumulation of glycogen and fat in the liver. So this result is producing symptoms of hypoglycemia, hyperuricemia, followed by hypolipidemia and lactic acid epidemia and hyperalanemia. And let's study the abnormalities in glycogen synthesis enzyme. Glycogen synthesis enzyme basically involved in the synthesis of glycogen. So this is the schematic diagram that we have already studied, the production of UDP from glucose. And then UDP glucose will serve as a glucose transporter to glycogen, and sequentially 8 glucose molecules will be attached and to produce a nascent glycogen chain. Okay, so we have already studied how glycogen is synthesized. Okay, so glycogen synthesis deficiency is inherited in an autosomal recessive manner, and it is a rare abnormality of glycogen metabolism which causes moderately decreased glycogen stores in liver. And symptoms typically begin in infancy or in early childhood and may include drowsiness, sweating, lack of attention, fasting, hypoglycemia associated with hyperketonemia, seizures, and other findings. Okay, so here are some enzymes involved in glycogen synthesis. So the abnormalities of those enzymes leads to the glycogen storage disease of humans. So here is the summary of interaction of glucose metabolism pathways in animals. So I have already discussed it in the class. So what happened, this is the source of glucose from glycogen or starch or lactate or amino acid or glycerol from non-carbohydrate source. This is the process of gluconeogenesis. So in the first step of glucose is produced pyruvate, then water and carbon dioxide, and then second step is pentose phosphate pathway, and after last, and this is the major two pathways of glucose metabolism. Or as we already studied, in anaerobic respiration, glucose might be converted to lactate and ethanol. So this is the schematic diagram to show the summary of interaction of glucose metabolism pathway in animals. So this class is ended here. Thank you very much.